Now I am going to explain the crossing over. It is a biological process in which exchanging of genetical material between non-sister chromatids of homologous chromosomes. As a result, it produces new combination of genes. This term was coined by T.H. Morgan in the year 1912. Especially this type of crossing over seen in meiotic phase. During the prophase 1 of Pachytene stage, crossing over can occur. Crossing over is divided into two types. They are mitotic crossing over and meiotic crossing over. Mitotic means somatic cell. The occurrence of mitotic crossing over is very rare. And second type is meiotic or germinal crossing over. And this type of crossing over, I already said, occurs in Pachytene stage of prophase 1 of meiosis, especially during the gamete formation. So it is also called, the name is germinal crossing over. Just observe the pictures. Here, two homologous chromosomes representing the different traits. Structurally they are looking alike, but they are not the same chromosome. They are different chromosomes. These are the two homologous chromosomes, one representing in the purple color and another in the light blue in color. And as a result of crossing over, we can get 4 gametes. Out of the 4 gametes, 2 are the parental trait and 2 are the recombinant. So the exchanging of chromosome between the homologous chromosome here or non-homologous chromosome. Both the chromosomes are different in nature. So we have to call the name as exchanging of chromosome between the non-sister chromatids of homologous pair of chromosomes. And next we are going to see about what are the mechanisms of crossing over. Crossing over is a process which includes the 4 stages. They are synapses, tetrad formation, crossing over, and last phase is terminalization. Let's see, the first phase is called as a synapsis. Synapsis is a process in which pairing occurs between the 2 homologous chromosomes. Pairing occurs between the 2 homologous chromosomes. Both the chromosomes are looking alike is called as homologous chromosome. During the phase is called as a zygoteny of prophase 1 in meiotic phase 1. So here 2 chromosomes, one chromosome representing the blue color and another representing the red color. Just observe the genes which is present in the chromosome. In the blue color, capital A and B's are present, whereas in red color, small a and small b's are present. So homologous chromosomes are joined together is called as a first phase, synapsis. Again I am recalling the homologous chromosomes are arranged in a side by side. As a result, it is looking like a pairing. So this you have to call the name is bivalent chromosome or synapsis or synthesis. It is divided into 3 types based upon where they are pairing. They are classified into 3 types. First one is called as a procentric, in which pairing occurs in the middle of chromosome. And second type is pro-terminal. Here terminal means tip of the chromosome. So pairing of chromosome occurs in the tip of the cell is called as pro-terminal. And third phase is called as a random. Random means occurring at somewhere. Pairing occurs at the anywhere place is called as a random. So these are the 3 types of synapses. Again I am recalling, pairing of homologous chromosome forms the bivalent structure during the zygoteny phase of prophase 1 in meiosis 1. It is divided into 3 types. They are procentric, pro-terminal and random. And second stage is called as the tetrad formation. Tetrad means 4. So after the pairing of chromosome, each homologous chromosome, bivalent in nature, began to identical sister chromatids. This is nothing but the duplication of chromosome to be occur. So bivalent, due to the duplication process, it leads to the formation of 4 chromosomes. So we have to call the name as tetrad formation. So at the end of the stage, 4 chromatids are formed. So they are called the name as tetrad stage or tetrad formation. And next we are move on to the third phase is called as a crossing over. Just observe the metaphase 1. They are aligned in the central line. Whereas in metaphase 2, the chromosome is going to be exchanged here. So blue pairing of chromosome, the changing of chromosome capital A is joined with the small b. Whereas in red chromosome, small a is paired with the capital B. How it is occur? So this process is called as the crossing over, which takes place in the Pachytene stage. Here, non-sister chromatid, again, they are going to be exchange the chromosome. For that, they are crossed like a crisscross pattern or H-shaped structures are going to be joined here. After that, joining, breaking and rejoining of the two chromatids are occur. So that structure is called as chiasmata. Again, I am recalling ma'am. During this crossing over, non-sister chromatids of homologous chromosomes crossed together like a crisscross pattern or S-shaped pattern. After the crossing, breaking and rejoining of the two chromatids are occur. Such process is called as a chiasmata. And recently their study shows that synapses and a chiasmata formation forms a specialized structure is called as synaptonemal complex. So during this crossing over, synapses and a chiasmata helpful for the crossing over by a special structure is called as synaptonemal complex. So such type of complex is absent in Drosophila. So there is no crossing over to be takes place. If the synaptal complex is present, crossing overs to be occur. If it is absent, there is no crossing over. So at the end of the terminalization, this chiasmata move towards in the terminal region leads to the separation of chromosome. So terminalization after the crossing over going to be separation of chromosome is called as a terminalization. So at the end of the terminalization, we can get 4 progenies. Out of 4 progenies, 2 are the parental tribe and the remaining 2 are the recombinant. So these are the mechanism of crossing over. Again, I am recalling the crossing over structure. It is divided into 4 phases. They are synapses, tetrad formation, crossing over and terminalization. And next we are move on to what are the types of crossing over. Just observe the first picture. These are the 2 pairs of genes. They are the non-sister chromatids of homologous chromosomes. This is the normal chromosome, so there is no crossing over to be present. So the percentage is 0. How they are calculating the percentage? This is called as a RF. RF means recombination frequency. So what is recombination? So during the crossing over, at the end of the process, we can get the progeny. How many new formation cells are formed? That is called as a recombinant, which is divided by the total number of offsprings. And previously we saw the 4 offsprings are formed. Out of 4, 2 are the recombinant and 2 are the parental tribe. So how we have to write the recombination frequency? So recombinant is 2. Total progeny is 4. So 2 by 4 is equal to 50%. So in the previous diagram we saw the crossing over is 50% of crossing overs to be occur. Based upon that, it is divided into 4 types. They are no crossing over, single crossing over, double crossover and multiple crossover. Let's see the first one, no crossing over. Here as it is the normal cell, so there is no exchanging of genetical material. So recombination frequency 0 out of 4 progeny. So the percentage is 0. And second type is called as a single crossing over. Just observe the center part. Here one chiasmata alone is formed. At one location alone the gene is going to be exchanged. So they are called, the name is single crossing over. So as a result of single crossing over, we can get 2 recombinant. So RF value is 2 by 4 is equal to 50%. And double crossing over means formation of two chiasmata. Formation of two chiasmata. Just observe the central picture here. Chiasmata we can see in the two places. So they are called, the name is double crossing over. This double crossing over again divided into three types. They are 2 strand, 3 strand and 4 strand. If 2 strand means chiasmata occurs in the two locations. There is no recombinants are formed, so our F value is 0. Whereas 3 strand double crossing over, chiasmata we can see in the 3 region. In the center 2 part and the down location also 1. So as a result 3 location crossing over to be occur. So we have to call the name as 3 strand crossing over. As a result we can get 2 recombinant. So the percentage is 50. And 4 strand double crossing over means 4 places the crossing over going to be occur. As a result we can get 4 recombinants. So 4 by 4 into 100 is equal to 100 percentage. So these are the double crossover. Again I am recalling if the formation of two chiasmata means we have to call the name as double crossover. It is subdivided into 2 strand, 3 strand and 4 strand. And last one is called as a multiple crossing over. That means formation of more than 2 chiasmata, but the occurrence is very, very low. And these are the types of crossing over. And next you have to see what are the importance of crossing over. This is the important 5-mark question mark. Repeatedly 5-mark question comes from this lesson. One of the important 5-mark question is crossing over, and 3-mark question is importance of crossing over. So listen carefully and learn well. Crossing over involved in both prokaryotic as well as eukaryotic organism. For example, bacteria, fungi, yeast, higher plants and higher animals. What are the importance? So this crossing over leads to the exchanging of chromosome. As a result, new genes or new combinations are formed. This plays a very important role in the evolutionary process. And the crossing over helps in the arrangement of genes in the chromosomes. So where the chromosomes are arranged, we can see by the help of the crossing over. And third point, this crossing over helps in the formation of genetical map construction. Next class I'll explain what is genetical maps. And this is also helpful for the understanding the occurrence, nature and how the mechanism of actions to be occur in the places. And last one, it is very helpful for the plant breeding purposes because of the exchanging of chromosome, new combinations to be occur. So these are the 5 important process of crossing over. Helps us for the new organism, know for the where the genes are located, identify the genetical map as well as the identification of occurrence and nature of the gene role and various role in the plant breeding techniques. Thank you girls.